So before we cover proteins, let's look at how we use dehydration synthesis to put together carbohydrates, because the process is very much the same. And then when we reach that point in putting together amino acids and our information on proteins, we'll hop back to our drawing board here and sketch out how the process looks when we combine them together. All right, so for carbohydrates, we want to start with a monomer, a monosaccharide. And so if I'm able to choose whatever monosaccharide I want to use, I'm going to go to the one that's most often discussed when it comes to carbohydrates and simple sugars, the one that flows through our bloodstream to maintain proper blood sugar levels, the one that's produced in plants during photosynthesis, and that is the monosaccharide known as. So glucose, all right, so to sketch this guy down, we know that it's a hexose sugar, and we should just kind of remember that its structure is like a hexagon. So we'll draw like a simple hexagon starting out, like so. And we should recall that one of these corners is actually occupied by oxygen. So we put like a little O there to represent oxygen. And what we need to know to be able to diagram dehydration synthesis is where those guys will actually get the water ripped away from them and where that connection will be made, a new bond will be made. But we should also, I guess, remember that there's a little carbon that hangs off of this structure, and it's going to be with H2 OH. It's a CH2OH little flag that sits on top of our little hexagon boat, I suppose, or submarine, or whatever you want to consider this to look like. Because it is glucose, we know that it has hydroxyl groups that alternate. So that's what we need to actually sketch in, because those will be involved in our dehydration synthesis. So it really doesn't matter. I'm going to do one that's up first, and then we'll draw one that's down to the bottom, and then up in the structure itself, and then another one that's down here. All right, now as far as carbons, yes, you're right, this has 6 carbons. Right, so for our first carbon is here, we can kind of see that, it's obvious, but the other ones are shown in the structure, so we don't need to draw or write every single carbon out. So this will be carbon 1, and then 2, 3, 4, 5, and 6. So those are your 6 carbons. We should remember that carbon will make 4 bonds always. And so wherever a carbon is, and we do not see 4 bonds coming from it, we should make the assumption that hydrogen must be the other thing that's there. Right? And when you do that, again, to save ourselves some writing. But we don't need all those written in for these guys. We simply need to draw another glucose molecule to show how these guys will get put together. So I'm going to cheat and grab all this and clone it out. You'll have to actually, or I can twist it all over the place. That'd be good too.
Wow.
Yeah. And we'll set it up like this. Sure. All right. So we have 2 glucose molecules. Right. If they're going to be put together by dehydration synthesis, what bits of each one will be where the bond occurs? Where will dehydration happen? Right there in the middle.
Like that one?
Uh-huh.
And then right up there.
This guy?
Yeah.
Okay. All right. Yeah. Yeah, so these 2 hydroxyl groups are going to be what works to form ultimately the bond between these 2 by dehydration synthesis. So what gets removed from these guys? Because dehydration, we're removing water, H2O. So we're going to rip that H2O out of there. All right, now remember, we have to work with what we're given. We can't add extra things or remove more than what we have. So we know 2 of these hydrogens will be used for water, so those go away. And 1 of the oxygens will be used. So we'll just use that guy. Right? What's left? There's still an oxygen hanging out there yet. Right? And that's where that bond is going to happen. Right? That's where that glycosidic bond, it's a special name for a covalent bond where we're going to share electrons. And it's going to happen between this carbon that we just removed both the O and the H from, and this oxygen that's still left remaining on the other glucose. So let me go ahead, I'm going to erase out what we've used. So we used that, and I'm going to erase this guy because I'm going to move him a little bit. So this oxygen is still hanging out there like so, and it's going to bond here. So this is that glycosidic bond that's formed between two monosaccharides when we go through dehydration synthesis. Dehydration, we remove the water, and in synthesis, we put two things together. Right? Now, in the past, you've been given the diagram. Right? Now you should be able to, again, just with glucose, use glucose as your model monomer for carbohydrates because it just works really well. It's very common, and it'll serve you well beyond this class. Like if you decide to take AP Bio or whatever else, if you're going to go towards a medical career, this will be like helpful for you. Okay, so the idea here is that the proteins we're going to look at first the 8 major functions that proteins do. All right, we're going to look at those first. So we're going to make a list of 8 functions. We'll add that to the margin or the bottom or someplace you have room for your proteins. Then we'll focus on the monomer, the amino acid. There are 20 of them. We don't need to memorize them, but we're going to have to know the general format for an amino acid. We'll look at that. And then how they go from being by themselves to being put together to eventually becoming a protein. Right, so that's our roadmap to success with proteins today. Okay, so the types or major functions of proteins, sometimes noted as the classes of proteins. Right, but we can call them major types or functions as well. Right, the first type are contractile proteins, just like it sounds. Your muscles contract and relax to provide movement. That's what these proteins are. They are the proteins that make up your muscle fibers are contractile proteins. Right? So consider or think of movement of organism or even just movement of muscles, like muscles themselves are made up of contractile proteins. And of course we have like just fast and slow twitch muscle fibers and stuff like that. So there's a wide variety of these contractile proteins, but they fall underneath the umbrella of being contractile proteins. All right. The second type would be defensive proteins. So think your immune system, think defensive, like trying to protect yourself from infection. So antibodies and antigens. And so the image that I have off to the right here, this green glob here is being kind of picked on by these antigens, these defensive proteins. They're trying to find out which one of them has the right receptor to latch on to the outer part of that invader and start to break down either his cellular membrane if it's a bacterium, or if it's a virus to render it inert, I suppose, or unable to function. All right, we also have a third function known as enzymatic proteins, or enzymes for short, right? So these enzymes are catalytic molecules, meaning that they're going to help chemical reactions happen, or maybe even allow them to happen at a quicker pace, because they're going to reduce the amount of energy needed. The last part of Unit 2, we look at enzymes specifically because they are a very crucial, critical part to our own existence. When we eat, we have certain enzymes, these proteins that serve a function to help us digest things. And what's interesting is if we did not have those digestive enzymes, what normally would take us about 12 hours or so to digest, like a meal, with those same chemical processes would take about 70 days to complete. Right? So these enzymes are like hugely, critically important when it comes to us existing. If it took us 70 days to digest a single meal, then we would be like more along the lines of like a fungus or something. We would not be very mobile. We would not have a lot of energy to waste or expend. We would just be very slow-moving creatures indeed. So it's nice that we have those enzymes. And then fourth out of the 8 functions or major classes would be hormonal or regulatory proteins. Right, so these are going to be things that control physiological processes. So like your insulin would be a regulatory protein. It's that gatekeeper to allowing glucose in or out, or into the cells, or keeping it out. The fifth function or class would be signal proteins. Okay, they're going to, just like it sounds, they're going to receive and respond to signals given off by other molecules or other proteins like themselves. So if you're thinking that there's a connection between this and whether you need insulin or not, that's correct. Right? Your body's not just going to dump insulin whenever it feels like it. Right? It's going to actually have some sort of feedback pathway that says, "Oh, dude, we need some glucose. Fire up the insulin. We need that." Whoa, we have way too much glucose here. Shut it down. We don't need any more. We need to be able to control that those regulatory proteins, and we can do that through signaling. Right? So that there's some proteins that's our job, just to simply either encourage things to work or not to work, or stop or slow down or speed up or whatever. We have storage proteins, which will store amino acids, so more monomers to build more proteins if we need to. We have structural proteins, which will help support our own framework. So we're looking at tendons and ligaments, and even the thread of spider silk. That's a structural protein. And then also transport proteins, things to move objects around. So like taxicabs, to move things in and out of cells or in and around our bodies. I included in the bottom right, I should probably blow this guy up a little bit more, but that's hemoglobin, right? Hemoglobin is found in your red blood cells. It's several proteins combined together to form a super protein, this globular protein, and it helps you again move oxygen and carbon dioxide and other things around your body and your bloodstream. Also, we have proteins that help move things into and out of cells themselves, because there are some things that we allow just to passively move through our cell membranes, because we want to maintain equal hydrostatic pressure, so water in and water out. We don't really control that a whole lot, but maybe certain things we want to get into the cell to use, or we have certain things the cells have made and we want to transport those out of our cell when we get the right signal to do so. And so there's some proteins that wait for that signal and transport things across the membrane. Okay, so let's look at the monomer then for proteins, and that would be amino acids. Right? There are 5 basic parts, and this is found on your outline. The only thing I may have done is reorder it a little bit, but still 5 basic parts. Right? You're going to have a central alpha carbon atom. And we're going to say it's an alpha C or alpha carbon because it's going to be considered to be the first carbon we look at. It's the center of the whole complex that makes up an amino acid. All right? It'll have a single hydrogen attached. All right? We'll consider that. All right? So let me highlight that. This is our central alpha carbon with a single hydrogen attached. We then have an amino group. This is found on the left-hand side of our image. It's nitrogen with 2 hydrogens, so NH2. To the opposite side of the amino acid, we have a carboxyl group. We've seen this guy before. All right, our COOH, our COO or COO. Right? We saw that also with fatty acids. Right? We have a fatty acid was composed of a hydrocarbon chain that made it fatty, and then a carboxyl group that made it an acid. So it's the same idea here. An amino acid is an amino group, and it's an acid because it has that carboxyl group on it. Right? An amino acid. The R group is known as a side chain, or it's what makes one of the 20 different from the rest. Right? It's like a plug-and-play spot. All amino acids will have these items here. What makes them different from one another is what you connect here at the R chain. Right? So they'll all have amino groups. They'll all have that central carbon with a hydrogen. They'll all have that carboxyl group. It's just this R chain is different for each of the 20 of them. All right, so pretty picture, kind of pretty picture.
It's beautiful.
Thank you. All right, so we still have our amino group here, our central carbon, and our carboxyl group. Right, that's still there, and then this is the R group that can be interchanged with different things to make the 20 different amino acids. Right, so we do not need to memorize the 20 amino acids. What you do need to understand and what you do need to know is the basic parts that make up all of them. Right? All of them have an amino group, all of them have that central carbon, all of them have that carboxyl group. It's the R group that changes their name. Right? So, for example, if I wanted to consider the amino acid glycine, then I would remove the R group and its R group would be just simply hydrogen. Right? This would be the structure than for glycine. But we don't care, again, about memorizing names. What we need to know is that amino acids have that amino group, that carboxyl group, that central carbon with one hydrogen on it, and then whatever the R group is, that gives it its special name. Right? If we do another example, like alanine, right? Again, amino group, central carbon with hydrogen, carboxyl group, and then we replace the R group with that, with CH3. Right? Again, we don't need to memorize the names. We don't need to memorize the R groups. We just need to recognize that R groups are interchangeable between all the different amino acids. And then finally, one more. Again, there are only 20 amino acids. Each one just has that different R group. So if we want to think of like valine as another amino acid, then we replace the R group with whatever it is. Right? So they can be a little bit more elaborate and complex depending on what amino acid you're looking at. Do not memorize the 20 amino acids. But when you need to know, right, here are the 20 in all their glory. Here's what you absolutely should understand. Right? We're going to use these amino acids and we're going to put them together in a polymer, a long chain of them. And what makes them special is the R group that's attached. And different R groups have different properties about them. And so there are some amino acids where the R group makes it nonpolar. So it's water-fearing. Right? We have some that have R groups that are polar. So they're hydrophilic, water-loving. There are some amino acids that have a positive charge on them. There are some that have a negative charge on them. But here's the main reason why I'm sharing this, or why we include this with our information. When we put these amino acids together into a chain of them, it's going to cause them to bend and fold and shape themselves in a certain three-dimensional structure based upon whether they like water or hate water, have a positive charge or have a negative charge. Because if I have a couple amino acids that have positive charges near one another, they're not going to want to hang out near each other. They're going to spread out and away and bend away from each other. If I have some of them that are also negatively charged, then obviously the positive ones will want to lean over and be close to the negative ones because opposite charges attract. The water-fearing guys will want to hang out together. The water-loving guys will want to hang out together, especially in an aqueous environment, a water environment like the cell membrane, like within the cells, or your bloodstream, or whatever. So that's the takeaway here, that the different amino acids, the different R groups, cause them to have these different abilities or properties, and that will ultimately decide how, when we make a long chain of them, they're going to end up folding up. Okay, so these different R groups do that. Notice every single one of them has your amino, your carbon, and carboxyl. So if you have this on your screen, you can see this up close. You can kind of look through and see how we make proteins then, right? We're going to make them from the amino acids. The polymer then is going to be the protein that results. There are only 20 amino acids. But remember, it's the sequence of those amino acids that determine what the protein will end up looking like. Like whether it will curl up on itself, like upwards or downwards or sideways, or in helixes or in sheets or whatever. And so this is in your note set towards the bottom, I think, for proteins, I think. But there are only 20 amino acids, but there are thousands of different proteins. Like if we consider a single amino acid, now I put a name on it, it's leucine, but the name doesn't really matter. All we need to know is that this is a monomer, this is an amino acid, right? Well, our DNA is going to have that information, that instruction, the order on how we put these guys together, like which amino acids go where. So we have a DNA strand that opens up. We build an RNA strand. That RNA gets transferred to a ribosome. A ribosome says, "Okay, we got it," and they start building these amino acids in sequence, in order, right? And so we build a protein on a sequence of different amino acids, different monomers, and this sequence results in a certain shape that folds up when it's made, and it becomes a protein, a special protein that does a certain function. Right? We just call it protein A. If we take a very similar sequence, and even if, like in this one, I simply took like the leucine and the valine and I switched them around, the rest of it's the same, it still is going to be a completely different protein because of that change, that one change in its sequence, it's going to bend in a different way. It's got a different shape to it, slightly. and that's going to make it have a different function altogether. So there's a different protein, protein B. So if the sequence is different in any way, it's going to be a completely different protein. It even can be made of fewer amino acids, and that's going to be a different protein. All right, now this diagram has a lot going on, but it's not that complex. So let's at least try to simplify it, and then we're going to go back to our drawing board for a second. The question then would be, like, how do we get these amino acids to link together? How do we take one monomer and put together with another one and another one and another one and build our chain along the way? Right, so at the top of our diagram here, and again, if you haven't pulled up on your Chromebook, it's easy to kind of follow and see. If you're far away, it's kind of squinty. We have two amino acids. Okay, so we have, this is our amino group here on the left, our central carbon with an R group, and then a single hydrogen, and then we have our carboxyl group. And then on the other one, we also have an amino group, central carbon, R group, and then our carboxyl. So they're just like lined up. In order to put them together, right, it's going to be dehydration synthesis again. So we're going to remove a water. So these guys that are close together, the oxygen and a couple of the hydrogens are going to, well, we should have an OH here, but in this diagram it's not showing that. It's showing a charge instead. But those are going to get removed and used to make H2O. So we rip a water out of it, and then what's left? Well, we've used hydrogen, two hydrogens and one oxygen, so nothing's left other than to connect them from carbon to the nitrogen. So the carboxyl group on the first amino acid will form a link with the nitrogen from the amino acid, or from the amino group of the other amino acid. Right? And that's the start of our polymer. That's the start of building our peptide chain, or our polypeptide chain. So this is known as a peptide bond because of what it's making, like what we're putting together. But it's basically a covalent bond. It's just given a special name, right? We're sharing electrons to make this happen. And there's a lot of other steps that go into getting to this point of putting them together, but this is the basic. At the end of the day, we're removing a water and putting them together. Right? If we want to continue that chain along, we bring another amino acid in. It has an amino group, has a carboxyl group, and we're going to continue the process along to continue to build more and more as we move along our chain. Right? So we're gonna hop out of this and we're going to go into diagramming a little bit. So wherever you put that diagram of dehydration synthesis with carbohydrates, next to it you might want to put this process that happens with amino acids, because I think it'll help you like make a quick comparison. All right, so what we need to do is we need to be able to sketch roughly an amino acid. We just talked about the 5 parts that it must have. Right, so what are one of those 5 parts? Okay, the central alpha atom, or alpha carbon, right? So we'll put a carbon up here, and then it's always attached with one hydrogen. You got it. Do you want the hydrogen above or below? Okay, I mean, it doesn't matter, but we'll put it down here because we saw it there first, I think. Okay, so we have 2 of our 5 parts. What else? Owen?
An amino group?
An amino group, right. Where do you want that, left or right?
Left.
Okay, and then what makes that guy up?
Nitrogen, 2 hydrogens.
You got it. So nitrogen here, attached to the carbon, and we're going to branch off this guy with 2 hydrogens. Just like that. Okay, so we have our amino group. What else? Gavin?
A carboxyl group.
Carboxyl group, okay. And where do you want that guy?
Right.
To the right side. If wherever you put your amino group, okay, it's going to be opposite of that, from your central carbon. So, okay. And then what makes the carboxyl group up?
2 oxygens, a hydrogen, and a carbon.
Okay, so we put a carbon in to put it along our central little chain here. Perfect. We double bond to one of the oxygens. We single bond to an OH. Right? We've seen this before, again with amino acids, or with fatty acids. And we see it here again with amino acids. Okay, so we have we have 4 parts, 1 left. What's the last part? Dave. The R group, yeah. I'm gonna put this bonded like so. I'll put a big R here, and I'll put this R in a box to remind me that this is some special grouping of atoms. It makes each amino acid special. Right? So this is representing any amino acid. Okay, so far so good. Well, we need another one of these. So I'm just gonna highlight this guy, clone one out, and I'm gonna drag him over. And that'll work, sure. All right, so you guys have to actually sketch one out. Sorry. All right, so you gotta make another copy of what we just sketched out. All right, this could have a hydrogen on the end, so that make it glycine or whatever it is. We could put like CH3 on this one. It'd be another amino acid. Doesn't matter. They all have amino group, central alpha carbon with a hydrogen, and a carboxyl group. If we're going to build them into a chain, we're going to line them up. Amino, central carbon, carboxyl. Amino, central carbon, carboxyl. All along the way. Right now, if you were a betting person, all right, if you were to make a wager, where do you think, if we're going to put these guys together by dehydration synthesis, what would be a very convenient spot for them to build that connection at? Miss Penny.
The oxygen, hydrogen, hydrogen, are right by each other.
Yeah. Why don't we go after these guys right here? Because it seems to have everything we want and no more, no less. It works out rather nicely. And you're correct. That's where this is going to work. So, if it's dehydration synthesis, what do we end up having to do here? We're going to pull out all together, we're going to pull out H2O. So this whole thing, we're going to rip out a water. Okay, water gets ripped out and removed. Okay, is there anything left behind in what we've circled anyway? No, nothing left. So all this gets used up. So I'm going to just erase it, if that's okay. And then, where would be a good spot for these guys to link together? Go ahead, say.
Maybe the oxygen, hydrogen top.
No. Where did we just take things away from? We just took things away from this guy and this guy. So they're wanting to get some electrons from something. They're going to want to fill in the gap that has just been made on them. So where's it going to happen, Coswell? Between which what? You got it. Yeah, absolutely. So the new bond will form here. Okay? This bond is known as a peptide bond. Peptide bond. That's for bonus points, folks. I mean, that's like for the gold star for the day. If you can remember that it's a peptide bond, that's great. But it's a covalent bond. It's sharing electrons. Right? We have three things to finish up with proteins. Here's the first. We go from our monomer, the amino acid, right? We start putting them together by dehydration synthesis into a peptide, or sometimes people call this a polypeptide because there's many peptide bonds going on. So poly meaning many. And then to become a protein, and in my opinion, and probably I'm sure Mrs. Elster agrees as well, is that it becomes a protein when it actually wraps up and forms its three-dimensional structure. When all of those amino acids with like their polarities and positive and negative charges and water-loving, water-hating ideas, cause them to form a certain shape. At that point, they can do a function. They can serve their purpose. Right? Before then, it's like Power Rangers, right? Okay, like this is like your 4 Power Rangers, okay, and then they get into like their Zords, okay, but they can't really serve their purpose. Like ultimately, like wipe out the nasty until like they form into the Megazord, right? When they actually get into their final form.
Please define.
Pterodactyl, Triceratops, right? Saber-tooth tiger, three-toed sloth. No, that's not one of them.
Why am I so confused?
Right. But so that final form, that final shape, is what makes it a protein. That final form of all the Zords put together to form the Megazord is what is the final ultimate evil butt-kicking machine, right? Unless you add the fifth ranger, then you get like the special extra like head armor, shoulder armor, and blade of justice. But you know, or warhammer of death, whatever you want to throw in there. Okay.
The roar.
All right. So that's what we're talking about here. That we are not a protein officially until we actually form that three-dimensional structure. When we go from the sequence of amino acids to ultimately being a protein, it takes several stages to get there. Okay, so the first stage is known as the primary structure. And this is something that you might want to sketch off to the side or sketch in whatever you want to put. But so primary structure or first structure is simply the polypeptide. Right? We make this long strand of amino acids. Right? Now, the analogy is it's like the letters of the alphabet. Okay, so we put all these alphabets together, we put all these letters together in a certain sequence, right? We start putting a bunch of letters together, and some of these letter sequences actually seem to make words for us. And that's what happens with these polypeptide chains. Certain sequences will want to start to form into straight, flat sheets, and some of them will want to wrap up in a spiral like a helix, right? As this thing's being built. That's known as the secondary structure. Right? So it's like the ordered arrangement or conformation of these amino acids actually form special shapes just based on them being like in a certain sequence of themselves. Just like certain letters in the right sequence will be like cat or dog or fig or mugs or whatever. Is that okay?
Yeah.
So it's like stage 2. All right. The tertiary structure is where all these words, they're not like maybe in the right order. If we wrap them in a certain way, if we rearrange them in a certain way structurally, we can form them into a coherent sentence. And that sentence has some meaning behind it. And so that's what's going on with the tertiary structure. We get all these like different sheets and helices and the remaining bits of the peptide chain, and now because it's out in the environment, it wraps up on itself and forms a certain structure. Right? And that gives it function. That gives it purpose. It's a protein now. Right? All proteins do this. All of them. All of them have primary, secondary, and tertiary structure. Some of them require more than one protein to be combined to actually do their job. And that would be the quaternary or fourth structure. Right? It's like if we take several sentences and we get them together to make a paragraph. And that paragraph has meaning and a purpose. So those are your 4 main structural builds as you move from polypeptide chain ultimately to your protein in tertiary structure. And if you need to, like hemoglobin, needs more than one protein to be what it is in your red blood cells that carry oxygen and carry carbon dioxide and other things in and out of your cells and in the bloodstream and vice versa, right? So it would have quaternary structure. Okay. All right. So what does that mean at the end of the day? It means that shape absolutely matters. We talked about that at the beginning of proteins, that the structure of molecules really dictate what their function. Form determines function. It's very much the true idea here. Right, and so I picked up a few examples like growth hormone, pepsin, hemoglobin, collagen, and these are all proteins. Some of them are just single. This would be like a tertiary structure because there's only one, protein or one protein kind of all wrapped up on itself. Hemoglobin is several different proteins working together, so that would have a quaternary structure to it. But different shapes equal different jobs. That's really the takeaway. All right, so if shape is that important, if the shape that they wrap themselves into determine what they do and how they do it and if they can do it in the first place, anything that happens to these guys that changes that will ruin what they can do. Right? Like hemoglobin has to be like wrapped up in this format or else it does not do its job. It can't. So this is what we mean by denaturing. Right? When a protein is denatured, it means that we have unfolded it or modified its shape. Right? Now, this can happen in a couple different ways. It can be done by overheating it, right? So putting it into a warmer environment than what it originally has been formed in, or changing the environment that's in altogether, like the chemicals that it's with. And even, I guess we can include in there like the pH. Like there's some proteins that work well at neutral pH. Those are proteins that form and function within your cells, within like a typical cell. You have other proteins that work best with very low pH, like your digestive enzymes. They have to be at a pH of like 1.7 or whatever it is, super low for them to function. They function best, and that's why you find them there, because they function best in that environment. If you would take maybe like insulin and dump it into stomach acid, it's not going to function anymore. It's going to ruin it, because now it's outside a range of pH that it can actually work in. So why does it break it? Because it ruins the structure. What normally would behave like water-fearing and water-loving and have positive charges and negative charges that will attract or repel one another, that the environment changes that. Those either attractive or repelling forces don't work so well anymore. And so what normally was a good structure that could maybe carry oxygen or be the gatekeeper for letting insulin in or letting glucose in or not, no longer can do that because its shape has been changed. It's like you have a key for a certain lock, right? If you take a hammer to that key and pound the crap out of it, it will no longer work in the lock. You've denatured that enzyme. It can't unlock things anymore. It cannot function anymore. All right, now sometimes they can be repaired if you get it back into its normal environment. Sometimes it cannot. We're not going to worry too much about when it can or cannot. We should be aware that it's possible to ruin it to beyond repair. And sometimes it's not. If we get it back to its natural environment or where we normally would find it functioning best, it will still continue to work. It'll